Hello, welcome everyone. This is Simon Fraser University's Café Scientifique, and you are joining us today for a session with Dr. Ben Ashby from our Department of Mathematics. Thank you so much for joining us tonight. At this time, we will give it a couple more minutes as more people trickle into the attendee room. My name is Cynthia Henson, and welcome. I see lots of familiar names on the attendee list. Thank you so much for joining us again tonight. And there are a number of new names we're seeing for tonight. Welcome. Again, you are joining SFU's Café Scientifique, and my name is Cynthia Henson. We'll give it about a minute more for more people to come into the attendee room. All right, I guess we should get started. Again, this is Simon Fraser University's Café Scientifique. My name is Cynthia Henson. I work with the Faculty of Science, the Dean's Office, and I look after the Outreach and Engagement Program on behalf of the faculty. Café Scientifique is a discussion series where we feature different faculty presenters, researchers, to help us engage the public in their research work. We do this about 6 or 8 times a year, and we're staying virtual for now. Just a quick reminder, we are recording this event, and we will post the recording on our faculty website after tonight. There will also be a feedback link shared with all those who've been able to come to this event tonight, and we look forward to you sharing your feedback with us. Your emails have also been included in our mailing list, so you get to receive updates on the future cafes after tonight. It's great to see lots of familiar names. Thank you so much to all the regulars who come join us each time we do this. And a warm welcome to all those joining us for the first time. We also have a few people coming in from beyond BC. Thank you so much for joining us tonight. Before I proceed, I'd like to do a virtual land acknowledgement. Let me begin by acknowledging that we are privileged to gather today on the unceded traditional territories of the Squamish, Tsleil-Waututh, Coquitlam, and Musqueam peoples. We thank them for having cared for these lands and waters since time out of mind, and look forward to working with them in partnership as we continue to build this great city together. If you know whose territory you're currently on, please take a moment to reflect on this. And if you don't know whose land you're on, I encourage you to look it up and make yourself familiar with the indigenous peoples whose land you occupy. Before we proceed with our presentation, I'd like to give a few house rules for this cafe. There is a Q&A box that you have access to. We welcome questions or comments that you might have for tonight's speaker. We will try our best to read out everyone's questions after the presentation. You will note that the chat page is turned off, but you do have access to a Q&A box. The live transcript has been turned on for your convenience if you find that helpful. And again, as I mentioned, this event is being recorded and it will be shared broadly after tonight's event. Thank you. So at this point, I'd like to introduce our speaker for tonight. His presentation title is Going Viral, the Mathematics of Infectious Diseases. Lots of questions that he'd like to tackle for tonight. Why do some infectious diseases cause large outbreaks, while others never really take off? And what do infectious diseases have in common with memes? And where do all these COVID-19 variants keep coming from? So tonight we have Dr. Ben Ashby. He's a mathematical biologist interested in infectious diseases and evolution. He uses mathematical models to understand how infectious diseases spread through populations, how they evolve, and how in turn they affect the evolution of their hosts. He's an associate professor in the Department of Mathematics at Simon Fraser University. So please help me welcome Dr. Ben Ashby. At this point, Ben, I invite you to screen share your presentation. Thank you.
Hopefully you can see that. Ooh.
Yep, we sure can.
All right, thank you very much for the introduction, Cynthia, and for the opportunity to give a Café Scientifique, and welcome to everyone who's attending this virtually. So as you've just heard, I'm a mathematical biologist, and what does that mean? Well, that means I use mathematical models to understand things in the biological world, in particular infectious diseases. Now, I appreciate that many people in the audience don't have a mathematical background, and that doesn't matter. There's not going to be lots and lots of mathematics and equations in this talk. I'm going to be focusing on giving a high-level overview of the key concepts around the mathematics of infectious diseases and how we go about modeling them. So just from the off, don't worry if you're not really into mathematics, or you don't have a great understanding of mathematics, you don't need it to understand this talk. Okay, so to start with, I'm just going to give a bit of background about why I study infectious diseases and why I think they're really interesting. I think it's probably fair to say that prior to the COVID-19 pandemic, the idea of mathematical models of infectious diseases probably wasn't something that most people thought about. But given the last 2.5 years or so, we've seen that infectious diseases can sweep right across the globe and have a really severe impact on public health and the economy and society at large. And mathematical models have played a really important role in shaping public health responses, either trying to understand where an infection is going, or perhaps how new variants are coming into a population, and the kinds of interventions that we might take to try to combat infectious diseases. So, the idea of mathematical models, I think, is much more prominent in people's minds now when it comes to infectious diseases, as they've really shaped public health responses over the last 2.5 years or so. So as a mathematical modeler, I create models that try to understand how pathogens spread through populations. We can also make models to understand how pathogens evolve over time. And this little diagram here is demonstrating how these different variants with these Greek letter names, alpha, delta, omicron, and so on, have evolved over the last couple of years or so. So the more of a color that we have at a particular time point means the more common that particular variant is. So, in November 2020, most of this gray area were the original variant that we saw that came into the human population. We then saw the alpha variant come in, and then the delta variant, which swept most of the infections towards the end of 2021. And then around this time last year, a little bit later in the year, we saw the Omicron variants and then subsequent subvariants of Omicron as well. So we've seen this rapid pathogen evolution going on before our eyes, and obviously that has a really big impact on public health as well. So not only can we use mathematical models to try to understand how pathogens spread through populations, but also we can use them to understand something about pathogen evolution as well. Now, we tend to focus on infectious diseases of humans because we are humans, but infectious diseases, pathogens, and parasites are found throughout the natural world, pretty much wherever you look. If you're looking at other animals or plants, or even down in the microbial world, you find that there are host pathogen or host parasite relationships pretty much wherever you look. And it's one of those fundamental ecological relationships that's just like predators and prey, or plants and pollinators. They're everywhere that you look. And in fact, Jonathan Swift, back in the 18th century, came up with this little poem where he said, "So naturalists observe a flea have smaller fleas that on him prey, and these have smaller still to bite him, and so proceed ad infinitum." Really getting behind this idea that there are pathogens and parasites at all scales of life. Now, given their ubiquity and the often severe impact they have on host health and in turn host fitness, it stands to reason that lots of host defenses have evolved, whether again we're looking at our own immune systems or immune responses in plants, or even bacteria have defenses against viruses that they face. We see lots of different types of immune responses and defenses evolving to cope with the threat of different infectious agents. So, this is something we tend to think about if we think about our own immunity. And actually, if a paper came out in Nature last week looking at the evolution of immune genes associated with the Black Death, finding that there are certain genes that were selected for during that period. And so we're still seeing the imprint of past pathogen outbreaks on the human population. You also see this in sub-Saharan Africa with higher rates of sickle cell anemia, and that's been linked to higher malaria loads. So the idea there is that in regions that have more malaria, sickle cell anemia can confer some protection, or rather if you have one of the alleles for sickle cell anemia, but you don't have both of them. So we see the complex interplay between human evolution as well as pathogen evolution as well. And this leads on to ideas about how not just pathogens evolve and how humans evolve and other hosts might evolve with their pathogens, but how they're locked in a co-evolutionary arms race. And the reason I've got a picture of Alice from Alice in the Wonderland and the Red Queen here is because in Lewis Carroll's Through the Looking Glass, the Red Queen says to Alice, well, here you have to run as fast as you can just to stay in the same place. And the same kind of idea applies to hosts and pathogens, that hosts are trying to get ahead and try and resist their pathogens, and pathogens are trying to get ahead and infect their hosts, and they can never really escape from each other. So they're locked in this constant co-evolutionary battle. And we can extend these ideas beyond just thinking about things directly evolved in terms of disease and immunity. Think about other traits that might have evolved in response to pressure from pathogens. So if we go out and we look at, say, a peacock and has this wonderful bright plumage to attract mates, we might say, well, why do peacocks have these bright tail feathers to attract mates? Well, one possible hypothesis is that they allow females to identify males that are either resistant to infection or that simply they're currently not infected. And therefore, by mating with them, they either pass on good genes to their offspring, or they just have a lower risk of infection themselves. So maybe traits like this might have evolved in response to pressure from pathogens. We can also think again about other traits, physiological or behavioral, that might have evolved in response to the threat of infections. As in particular, sexually transmitted infections could have shaped mating dynamics as well. So who you choose to mate with and how often. So this, to link this all the way back to humans as well, we are not apart from evolution, and ideas about human monogamy may have been shaped by sexually transmitted infections. So one of the reasons why we're monogamous might be because of sexually transmitted infections. And our own human mating behavior will affect the evolution of sexually transmitted infections as well. So this woodcut here is illustrating syphilis in the Middle Ages. And when syphilis emerged in the Middle Ages in Europe, it was a very different disease to what we see now. And the German scholar Ulrich von Hutten said they had boils that stood out like acorns, from whence issued such filthy stinking matter that whosoever came within the scent believed himself infected. And then he went on to say, really interestingly, it was so horrible to behold that one would scarce think the disease that now reigneth to be of the same kind. And this is actually a really early description of evolution occurring, so pathogen evolution here. And within the space of a few decades, syphilis changed from being this really conspicuous, horrible, obvious disease to being one that was much more inconspicuous, and that allowed it to transmit better through the human population. So tying this all together, we have ideas about how pathogens spread through populations, how they evolve, how they affect the evolution of their hosts, and not just things related directly to disease, but traits beyond that, whether that's mating traits or bright plumages in peacocks, and so on. So what do I do as a mathematical modeler? Well, I try to understand all these types of phenomena using models. So whether that's looking at STIs and mating dynamics, or trying to understand patterns of host and pathogen diversity, to how we can intervene in pandemics, we can address all these sorts of problems using mathematical models. So what am I going to talk about today? Well, I'm going to start off by focusing on what is a mathematical model, and just give an overview of where do we start when we try to make models. Then I'll talk a little bit about what do models tell us about infectious diseases and other things that can transmit through populations like memes. And then finally, how do infectious diseases evolve? I'll talk a little bit about this towards the end. Okay, so first of all, what is a mathematical model? Well, I'll actually take a step back and ask, first of all, what is a model? Well, if I ask you what a model is, maybe you think of something like this, a model train set perhaps, or a model that a town planner might have. And these are physical models that are simplifications of the real world. Obviously, a model can't have all the complexity that we have in the real world, otherwise it would be the same size as the real world and have all of its complexity in it. So a model is just a distilled version of reality. And the same kind of principle goes when we come to mathematical models. They are a simplification of reality, trying to retain the key aspects. But we don't just have physical models, we also have conceptual models. And maybe you don't think about these types of models as much, but if we look outside and we see that it's cloudy, perhaps you think there's a 50% chance of rain based on your prior experience and how you understand the water cycle works. And you can make predictions from that model. Like I said, 50% chance of rain, or perhaps if you're in Vancouver, it's 100% chance of rain in this season. So we have these types of conceptual models as well, not just physical models. We can also think about how we interact with the real world. So here we've got Steph Curry shooting basketball, and given our intuition about how gravity works, about how the spin on the ball works, the speed at which it's released from your hand, we can come up with internal models in our heads of what the likely trajectory of that ball will be, and if you're going to make a basket or not. So maybe we don't think about these two sorts of things, the weather models and the models about when we're playing sports or interacting with other things, as models, but essentially they are models that we have in our heads that we try to build based on assumptions and our experience with the environment, and then we use to make predictions and how we therefore shape our interactions with the world. So what this means then is essentially we are all modelers, whether we like it or not. This is how we interact with the world. We take in stimuli, we make assumptions, we think about those assumptions, and we make predictions, and we act on those predictions. So we're all modelers. And why am I trying to hammer home this point? Well, at various points during the pandemic, and even prior to that, when it comes to things like models of climate change, people often say, I don't trust models. And what they mean by that is they don't trust mathematical models. But really, the mathematical models are just a formalization of what we do in our heads ordinarily. Okay, so if we want to come up with a model in our heads, what do? We see some sort of real world phenomenon, for example, an infection in a population. We make some assumptions about how that infection might spread, and then we come up with this informal model in our head. Okay, if you go near someone who sneezes, you might get infected. We think about that model, and then we come up with some predictions. Okay, if I stay away from this person, I'm less likely to get infected. And then we can interpret how what the outcome is. Was that prediction accurate, and do we need to refine our assumptions, our internal model? So this is a really simple process, but it's something that we do all the time. So what is a mathematical model? Well, a mathematical model is taking exactly the same process, but rather than just holding things in our heads, or in the case of model railway, putting it into a physical object, we're writing down those ideas formally. And when I say formal in quotes here, I'm really meaning mathematically. So by making formal assumptions, what I'm saying here is we write down rules about how our models will work, rather than just coming up with ideas in our head about, I have this idea about how infection spreads. I'm going to write down mathematically how I think it will spread. This then allows us to write down a mathematical system based on those assumptions, rather than just having something floating around in our heads. And then we can do formal analysis rather than just thinking. And the formal analysis is using mathematical rules to try to distill out some predictions. So these mathematical models are not a million miles away from the models that we use day in, day out without really realizing it. And so when you say you don't trust models, it doesn't really make sense, because these models here have the advantage of having all of their assumptions formalized and open to criticism, whereas the models that maybe you have in your head are potentially more open to bias. So hopefully I've convinced you that mathematical models aren't necessarily anything special. They're just a subset of the types of models that we come up with informally day in and day out. So you've probably seen this quote before, maybe, all models are wrong, but some are useful. This is a very cliche modeler quote that all modelers probably utter at some point during their lifetimes. What I want to focus on is, rather than all models being wrong, but some are useful, I want to focus on how are mathematical models useful, especially in the context of infectious diseases. So this word cloud here gives a summary of some of the types of things that we might use models for, or how they might be useful. For example, we might use them to fit to data, or to think about different public health initiatives. We might think about, is this scenario better than that scenario if we have this intervention? They can also, though, provide understanding and insights that we wouldn't get through the models that we just hold in our heads. And that's because often mathematical models reveal understanding about fundamental mechanisms, and they can give you some novel insights that are maybe counterintuitive to what you thought before. So our intuition can often fail us, whereas a mathematical model, because it's set in these formalized assumptions that are then formally analyzed, we can trace back and figure out why perhaps we get these counterintuitive results or predictions that we wouldn't have thought of previously. So there's a couple of examples of how models have been used during the pandemic. Say you want to know what's happening to the number of infections over time, and you've got some data points here. These black dots are just representing how many people were infected on a given day. And we can use a model to fit to these data. And when we're fitting a model to data, what we're really doing is we're changing the parameters in that model to try and say, okay, well, what best represents the pattern that we're seeing in the data using our model. Again, that model is based on some assumptions about, say, how transmission occurs. We can then perhaps project forward and then try to understand, well, what's it going to look like in the future? Is the number of infections going to go up? Is it going to stay steady? Is it going to go down? If so, how quickly? So we can do this sort of forecasting or projection into the future, and this has been something that has been used a lot during the pandemic. But there's always a lot of uncertainty in these things. So I've just presented a single line here, but you could think of these as three possible outcomes. Really, there would be a much wider range of outcomes, and so usually models will present a lot of uncertainty. So you might see that infections might be somewhere between 1,000 and 10,000. That's a lot of uncertainty, but maybe that is kind of useful because that tells you that you don't really know where you're going to end up, and there's a lot of uncertainty perhaps in how you're collecting your data. So there are different ways in which we can use models like this to try to understand what's going to happen in the future. Another way that we can think about the future using models is to think about how we can intervene into a situation. So you often see a do-nothing scenario, and I put this in quotes here because the do-nothing scenarios are sometimes poorly portrayed, especially in the media, because they're seen as worst-case scenarios and unrealistic. But really, these are kind of your null hypothesis, assuming that nothing is going to be changing in the population, and against that you can measure how different interventions will change things. So, for example, intervention A might be something like wearing masks, and interventions A and B might be wearing masks and encouraging people to work from home. So we can think about these different scenarios and the impact that they might have on infections based on our understanding of how infections occur, and then we could try to maybe recommend this scenario of interventions is better than that scenario. Okay, so the second section of my talk is going to be about what do models tell us about infectious diseases and other things that transmit as well, like memes. So really the first thing that we need to think about is, why do epidemics start in the first place? So why do they take off? And to have an epidemic taking off, we need three things. First of all, we need a pathogen to be the thing that's going to be taking off. We need some susceptible hosts that pathogen can infect. And then we also need there to be transmission. So just having a pathogen and susceptible host doesn't necessarily mean an epidemic will take place. We need to have a transmission process as well. Even then, that doesn't guarantee that we are going to get an epidemic. To understand whether we're going to get an epidemic or not, we need to know about the most fundamental quantity in mathematical epidemiology, which is something called the basic reproduction number, or R0. Now, many of you might have seen this R0 either in a different context, say if you're a biologist, or you might have seen it in the media. This number, although it's a very simple thing, it can help us understand some really powerful things about how epidemics take off, and also how we can potentially control them as well. What does this actually mean? It's a bit of a wordy definition, but it's the average number of infections caused by one infected individual in an otherwise fully susceptible population. So, like I said, that's quite a wordy definition, but really you can think about it as, if we have a population, suppose this is our population, all these blue dots is a different person, and those blue dots are all susceptible, so everyone can be infected in principle, and we're going to introduce one infected person who's indicated by this red dot here. And then all we're going to do is we're going to count how many infections they cause before they recover or die from the infection. So in this case, they've caused 3 infections. So our R0 here is just going to be 3. Now, this is very easy to do in a simple diagram like this, but when it comes to real data and real populations, it's much harder to estimate this R0 because we're not just introducing one person and able to accurately measure how many they infect. There's also lots of variation within populations. So, realistically, you'd want to take an average, and that's why we talk about on average, how many infections do we expect. So what does this R0 tell us? Well, logically, if this R0 is greater than 1, that means that early on in our outbreak, each infection leads to more than one new infection on average. So that means that the number of infections is likely to grow over time. So it's likely to take off. Whereas if our R0 is less than 1, then each infection leads to less than one new infection on average. And so our number of infections is going to decay. So this R0 tells us something really powerful based on whether it's greater than 1 or less than 1. And if we think about some different viruses here, if we look at, say, influenza virus, that has an R0 estimated between 1.2 and 2. These, incidentally, will depend on the population and the particular virus and the makeup of immunity at the time. Typically, we're assuming that the populations are fully susceptible. In practice, they might not actually be fully susceptible. Regardless, our influenza has somewhere of an R0 between 1.2 and 2. SARS-CoV-2, which causes COVID-19, initially had an R0 estimated somewhere around 3, but due to selection, later variants have much higher R0s. I put a question mark here next to 7 because there's quite a range of estimates here. One of the reasons that we haven't had flu seasons for the last couple of years really is because the interventions that we've taken have suppressed the R0 value for both COVID and influenza, and because influenza has a lower R0 than COVID, it's ended up being suppressed. That's why we've not really seen flu seasons. I've included chickenpox and measles down here at the bottom, which have much higher R0 numbers. These are both childhood infections, and the reason they're childhood infections is because they have high R0 values. So your R0 tells you something as well about the average age at which someone is likely to first be infected by something. In the case of measles and chickenpox, they have very high R0s, which means people tend to be infected early in life when they're children. It also means that it's very hard to control these sorts of outbreaks if they have much higher R0s. The reason being is if we vary our R0s, this little animation here is just showing the proportion infected as our R0 varies at the top, and we've got the time in days along the bottom. We see that when our R0 is very high, we get a very steep, fast epidemic and a high peak, whereas then when the R0 gets lower, it flattens out. We get a much lower peak, but we also get a longer outbreak as well. So all else being equal, this R0 can really change the shape of our epidemic curves. Okay, so suppose we've got an R0 greater than 1, what happens early on in an outbreak? Well, what we see is exponential growth of infections. So suppose we've got one infected individual here and we have an R0 of 2. What does that mean? Well, it means that they tend to infect 2 people on average. They each infect 2 people as well. They each infect 2 people. And so we have this doubling. And that's really the key thing about exponential growth. If we're to plot a curve of infections over time here, we'll see that early on in an outbreak, we have what's known as exponential growth. And this is one of the key concepts during the pandemic that I think has been really poorly communicated or misunderstood. Often people have confused the term exponential with fast. and they're not synonymous. So people have looked at this and said, okay, if we're down here at the early stages of this outbreak, maybe it's taking off, but it's not exponential yet. And they only say it's exponential when it starts getting steep up here. And you say, we've got exponential growth. Really, though, we've got exponential growth at all of these stages. It's not something that just suddenly occurs when it's fast. So, when we're looking at data and we see how cases are changing over time, we might see that, say, infections go from 100 to 200 to 400 people. That might be worse than if we're looking at 1,000 going to 1,100, 1,200, 1,400. And that's because in the first case, we're having this doubling, whereas in the second case, it's perhaps only a linear growth. So it's not necessarily about how quickly things are growing or how many you have in total. It's about the rate at which things are changing. So that exponential growth is really quite important. How does it matter in terms of something like disease outcomes? Well, if we imagine a scenario where here we've got a disease that causes an infectious disease that causes death, and let's suppose that there's a delay between infection and when someone tends to die. Suppose that it's a 3-week delay. Now, the problem with that is that if you take action in week 2, say, to try and reduce infections, the people that are dying in week 5 were infected already by week 2. And so here we've got the weekly deaths are doubling twice per week. So it goes from 1,000 to 2,000, 4,000 from week 1 to week 2, and then 4 to 8 to 16,000 between weeks 2 and 3, and so on. And so we see that if you've got an infection that has a delayed effect in terms of some measure, whether it's hospitalizations or deaths, and you're focusing on that measure as to when to act, really you need to be thinking much earlier on. So you have to be acting a lot earlier, because the costs grow exponentially if you wait. So what happens if you were to wait a week in this case? And this is a very simplistic example here, but we see that delaying by a week causes a massive increase in expected mortality by the end. So by the time your actions start having any effects, this final week here in week 6, we end up seeing an additional 1 million people have died in this made up outbreak here. So that's over 75% of the deaths occur just in this last week. And this is really the power and problem with exponential growth, is that delaying when you have exponential growth means that you have exponentially growing costs. And this is essentially what happened in many places. I was in the UK during the start of the pandemic, so my experience is really UK focused. Estimates in the UK are that if we had locked down a week earlier in March 2020, that we would have saved tens of thousands of lives, and our lockdown would have only had to last 5 weeks less than it did in the end. So for every week we had locked down earlier, I think we would have saved about 5 weeks of lockdown, not to mention tens of thousands of lives. So just 1 week earlier action can have a really significant effect later on because of this exponential growth. Okay, so what does this have to do with memes? I've mentioned memes a couple of times. Memes are, this term meme was coined by the evolutionary biologist Richard Dawkins in his book, The Selfish Gene. And he wasn't talking about infectious diseases then, he was actually talking about genes propagating through a population. And it's a very similar process to how infectious diseases spread. And he said, just as genes propagate themselves in the gene pool by leaping from body to body via sperms or eggs, so memes propagate themselves in the meme pool by leaping from brain to brain. So what is a meme? Well, a meme is something that has a cultural component and information that spreads from one person to another. And you might think of memes that are shared on social media, things like this, images or videos, or it could be things like challenges that are spread by word of mouth, things like fashion trends, or if someone whistles a tune and you hear that tune and you think that's catchy. The reason we say it's catchy is because it has this transmission like phenomena. So we say that things go viral because they behave like viruses. So what does exponential growth of memes look like? Well, it's basically the same process as we had for infections. If someone posts something on social media and 2 people share that, and then 2 more people share that, and 2 more people share that from each of these people that have seen it, we end up seeing exponential growth. So just in the same way as we saw exponential growth of an infection, we can see exponential growth of memes. So the tools that we use to understand infectious diseases mathematically, we can use to understand the spread of information in populations as well. Okay, so that's all about what happens early on in an epidemic? Why do they take off? And why do we see exponential growth of means? How about what causes epidemics to end? Now, we can basically do a similar process to what we did before, rather than looking at the R0, which was the average number of infections caused by one infected individual when the entire population is susceptible. Instead, we can look at something called the effective reproduction number, which is RE. And this is the average number of infections caused by one infected individual at a given time. So R0 is essentially a special case of this, looking at right at the start of the epidemic, whereas this RE number, it has different names. Sometimes it's just called the R number. This is taken at any point during an epidemic. And this is the number of infections that we expect one infection to cause during the course of that infection. And this is really important because essentially we can use the same logic we use with our R0 to understand whether an epidemic is going to keep going up or it's going to start coming down. So if this RE is greater than 1, that means that each infection leads to more than one new infection on average. And if the RE is less than 1, then each infection leads to less than one new infection on average. So if RE is greater than 1, we see infections will keep rising. And when RE is less than 1, where we're to the right of this dashed line here, we see the infections will start falling. So just as our R0 tells us something powerful about whether an epidemic is going to take off, our RE number tells us whether it's going to continue rising or whether it's going to start falling. So how do we work out this RE? Well, we could look at data. If we're doing a mathematical model, we can look at the key processes that come together to make this RE number. And what do we do to work that out? Well, we need to multiply together 3 things. Because we're interested in the number of infections on average that each infection leads to, what we need to do is we need to take the probability of transmission every time there's a contact between someone who's infected and someone who's susceptible. We multiply that by the number of susceptible contacts that they have per day. And we then multiply that by the time that they spend infectious. If we multiply all these things together, we get the average number of infections that each infection will produce at a given time. And so when we try to have some sort of intervention to bring an epidemic under control, we target different aspects of this RE number. So for example, if we use masking or try to increase hygiene, we can reduce the probability of transmission per contact. There are other things as well that could reduce this transmission probability, like seasonality. So the reason we see respiratory viruses in the winter tends to be because people spend more time indoors. We could tackle the number of susceptible contacts potentially by doing things like isolation, working from home, or reducing contacts between infected and susceptible people by doing regular testing. But this can also come down through the buildup of immunity in the population. And this has been something that's been quite contentious at points because of how people try to apply this principle. But the basic mathematical principle of a buildup of immunity in the population reducing this RE number is mathematically sound. It's more about how you get there that's the problem. You could also potentially try to reduce the time that people spend infectious by improving treatments as well, but I won't really focus on that here. Okay, so one thing I want to talk about here is herd immunity. And again, just like exponential growth, this is one of those things that I think has been misunderstood quite widely, and there are lots of common misconceptions about what herd immunity is and what its implications are for disease control. Really, herd immunity is tackling this number of susceptible contacts per day. So if we go back to our population here, so remember the blue dots correspond to individuals who are healthy and susceptible to infection. We'll have some grey individuals coming in. Those are people who are recovered and are immune, and the red dots are those who are currently infected. And we're going to assume to start with our RE number is greater than 1. So that means that each infection on average leads to more than one infection. And then if we allow this to start spreading through the population, we see that the number of red dots increases. And then as people recover, the number of grey dots starts to increase as well. And eventually the number of red dots starts to decrease. And the reason this decreases is because we are running out of susceptible hosts. So that means because our number of infections is falling, that means our RE number must be less than 1. So we're on that decline of our epidemic. And the whole point of herd immunity is that our RE number can be less than 1, even if we've still got susceptible people in the population. So we've still got some blue dots hanging around in here. And eventually, we should see all of the red dots disappear and everyone will be immune. And sorry, not everyone would be immune who had been infected. And we still have some people who are not immune. And all this then means is that any infection that comes into this population, RE number is less than 1 if we have sufficient immunity against that infection. But we don't have to have everyone immune. And that means that we have this indirect protection, but it's not perfect protection either. So there's lots of reasons as to why this has been poorly misunderstood, or poorly understood, I should say. One of the reasons is because we achieve herd immunity in a population when our RE number is equal to 1, and this is known as the herd immunity threshold. So even if we don't have something like vaccination, if we just let a population be infected and we have this epidemic curve like this, if we don't have any interventions, changing behavior or anything like that, we will achieve herd immunity when we hit the peak of this epidemic by its mathematical definition. However, that doesn't mean the epidemic is over. And we get all of these infections that occur to the right of that dashed line still. So your herd immunity threshold doesn't tell you how many people will be infected in an epidemic, it just tells you where the peak will be. It's also very different if you're allowing an infection to spread versus if you are doing something like a vaccination campaign for, say, childhood diseases. So the key point is where you're starting from. So in the case of a vaccination campaign, where you say vaccinating against measles regularly for young children, you have typically measles will be at a low frequency in the population. And so your RE number being less than 1 is about preventing an outbreak from taking off. Whereas if you just allow an infection to spread through a population and you reach herd immunity at this peak, your RE number being less than 1 is just saying that your infections are declining. So, but they're declining up here from a very high number. Whereas if you're vaccinating a population and you haven't had an outbreak in a while, that means that your RE number is less than 1 and you're starting from a low starting point. So herd immunity is really about this RE number being less than 1, and it doesn't really tell you anything about where you're starting from. And so vaccination and building up naturally acquired immunity can give you very different long-term outcomes. But the mathematical principle here of herd immunity still relies on the same threshold. So what is that threshold? Well, it turns out that in these very simple models, we can come up with a nice simple expression related to our basic reproduction number, R0, and that's 1 minus 1 over R0. So what does that look like? If we plot our basic reproduction number along this horizontal axis, and then the herd immunity thresholds, the percentage of the population that needs to be immune to prevent an outbreak, so for our R number to be less than 1, we see that we get this really steep incline to start with. So as we cross the threshold of R0 being greater than 1, we need some people in the population to be immune to prevent an outbreak. And this rises really steeply, but then eventually plateaus. So if we think about something like COVID-19, with its original R0, when it first came into the population of somewhere around 3, that means that you need somewhere in the mid 60% of people to be immune to reach herd immunity. But due to evolution, we now have variants that require much higher immunity in the population to reach this threshold because they have a higher R0 number. If we compare this to something like measles, the reason that it's very difficult to eradicate measles is because it has a very high R0, and that means that you need to maintain a really high herd immunity, really high level of immunity in the population to achieve herd immunity. So it's very difficult to eliminate something like measles because it has a really high R0 number, and that means that we need somewhere 92–95% of the population to be immune in order to prevent measles outbreaks. That means if you have vaccine scares, it's very easy for measles to cause outbreaks because you just start to dip below that herd immunity threshold, and then an outbreak can occur. And the reason that an outbreak can occur is because some of those people aren't going to be protected from infection. And they're only protected when we have sufficient immunity to prevent these infections taking off. Okay, so I mentioned evolution a moment ago about COVID-19 and how this has changed. I want to talk a little bit to finish up about how do infectious diseases evolve. So this is a key part of my research in terms of looking at trying to understand the key principles about how diseases spread, but then how do they evolve as well. So they can evolve various different components of their characteristics. One thing that say a pathogen might change is its transmissibility. So mutations could lead to different variants that have different transmissibility. So maybe one is more transmissible than another. And when I'm talking about this transmissibility, I'm talking about its intrinsic transmissibility. So this isn't necessarily dependent on the host. The second thing that might evolve is its ability to evade immunity in the population. So this is something that's been driving immunity. Immunity has been driving evolution for new variants of SARS-CoV-2. I'll talk about that in a moment. Something else that might change is the type of disease that it causes. So we might see, say, more asymptomatic disease, or it might start infecting the upper respiratory tract instead of the lower respiratory tract. We saw with the example of syphilis that the symptoms of disease can change due to natural selection. So there are different components that can potentially evolve and are under selection. I mentioned SARS-CoV-2 a minute ago, so this is the virus that causes COVID-19. Early on in the, when we saw the Alpha and Delta variants, we saw quite significant shifts in intrinsic transmissibility. But lately, the key thing that's been driving recent variants, in particular the Omicron and Omicron subvariants, has been their ability to evade immunity. So you can still see changes in transmissibility, you can still see changes in disease severity and symptoms, but really the key thing that's driving things at the moment is immune escape. And we didn't see this early on in the pandemic because there wasn't much immunity in the population. So, you know, transmission, strains like Alpha and Delta that had high transmissibility were advantageous, but then later on, the build-up of immunity in the population has meant that we're seeing variants that escape immunity. So the patterns of selection have shifted, and we can understand how those patterns shift using our models. So in principle, what happens when we're having infectious diseases evolve? Well, if we go back to our RE number, so if you recall, this is the average number of infections that each infection is producing at a given time. And this is the product of the transmission probability per contact, the number of susceptible contacts per day, and the time that an individual spends being infectious. Well, if we look at the transmissibility, a more transmissible strain is going to increase its transmission probability. If we have something that exhibits immune escape, it's increasing the number of susceptible contacts that it will be able to infect. And if something changes its disease symptoms, maybe it has higher level of asymptomatic people that are, when they become infected, they don't show symptoms, and therefore they can keep moving around and infecting people for longer. They could potentially spend longer being infectious. So the different types of characteristics of a virus or any pathogen indeed that can evolve affect these different components of its RE number. So to take a numerical example, suppose we have an original strain of a new pathogen and its RE number at a particular moment in time is 2, and that's composed of transmission probability of 1/10. Each person has 4 susceptible contacts and per day, and they spend 5 days infectious. So we end up multiplying these all together and we get an RE number of 2. And then suppose we have some mutations that lead to a new variant that has a different transmission probability. In this case, it's doubled its transmissibility. So it hasn't changed these other components, but it's changed this first part, its transmission probability. And because that's doubled here, we end up with an RE number that has gone from 2 to 4. So that means that the original strain caused on average 2 infections per infection. This new one causes 4 infections per infection. So this one is fitter in an evolutionary sense, and so will tend to increase in frequency relative to the other one. Of course, other components might evolve. So suppose we have immune escape. So now rather than having 4 susceptible contacts, suppose each person has 10 contacts a day, but only 4 of them are susceptible in the first case because the others are immune. If our RE number for our new variant is doubled, it could double because we have immune escape, and now rather than 4 susceptible contacts, we have 8 susceptible contacts per day. So this is one way in which the RE number can go up. We could also see exactly the same process in terms of doubling our RE number by having an infectious period that is longer. So maybe this is having a longer asymptomatic period, maybe it's having less severe disease symptoms. So a common misconception that we often see is that this idea that viruses and bacteria should evolve to be less virulent, less damaging towards their hosts, because it's not in their interest to kill their hosts. And to a certain degree, that's true. They don't want to kill their hosts. All they want to do is replicate. I use want in a very loose sense here. And this idea of, if they become less virulent, then they're going to increase their average time that they spend infectious, and therefore they should increase their RE number. So we should see selection pushing them towards lower virulence, because that will maximize this RE. However, that doesn't play out in reality. The reason being is that these levers, the transmission probability, the infectious period, which is linked to virulence, and even immune escape, these are not independent levers. As you move one of these levers, it can start shifting the other lever. So for example, if a pathogen starts becoming less virulent, that might be because it's infecting fewer host cells. And because it's infecting fewer host cells, it's producing fewer transmission stages. And so its probability of transmission decreases. Conversely, if it becomes more and more virulent, the reason it's doing so is because it's infecting more and more cells, and so it could increase its transmission, but that also reduces the infectious period. So as you start pushing one of these levers one way, it might start pulling one of the other levers the other way. As a result of that, we might see something more like this, where rather than virulence going higher and higher, or virulence going lower and lower and lower to maximize this RE number, it ends up at some middle level, okay? The crucial thing here is that we don't necessarily know when a new pathogen comes into a population, which side of the curve we're on. So we could be on the left-hand side, where actually selection will allow it to increase and become more virulent, or it could be on the right-hand side, where selection means it becomes less virulent. But we shouldn't expect that pathogens will just become less and less virulent over time, because this is not a pattern we tend to see. Okay, finally, just to wrap things up, I just wanted to mention this game that I have developed with a group in the UK called Then Try This, called Vaxcraft. If you're interested in understanding a little bit more about how viruses can evolve, I will just share my screen because this is an online game. I'll ask Cynthia to share a link to this game if possible. So you can go online at vaxcraft.thentrythis.org to play this game. And the idea here is that you've got a population of these little grey blobs, and you want to protect them using vaccines. But when you vaccinate the population, because you build up immunity in that population, that shapes the evolution of the virus. So the shapes that appear are shapes that you want to match with your vaccine. So here we've got all these blobs. This is our host population, and on the right-hand side, we've got a virus with lots of squares. So I'm going to vaccinate all these squares. We now see a new variant emerges, and it's still got all squares in this case. We also see that immunity might wane in the population, so that's why those squares disappear. When we see these green squares inside these hosts, that means that they're protected from viruses that have green squares. So eventually you might see a new variant emerges like this, and so, oh, now it's got some squares, it's got some circles. So that means that I need to start changing my vaccine. So it needs some circles in, maybe, it needs some squares in, and unfortunately I'm going to die because I've been walking rather than playing the game. But if you're interested in understanding a little bit more about how we need to, say, adapt our approaches to deal with how viruses evolve, please have a go at playing this game, and hopefully you will learn something and enjoy it as well. With that, I'll wrap things up. Thank you everyone for your attention, and I'd be more than happy to answer some of your questions.
Thank you. Thank you so much, Ben. I particularly enjoyed that Vaxcraft, and I'm happy to share that link if people are interested in that. Just send me an email, and I'm happy to share that link with everyone. So at this, yes.
Yeah, just quickly. So I'm also doing the Science Spectacular. I don't know if you're advertising this on here, but there's also a physical robotic virus. So I'll be at the Science Spectacular at SFU at the weekend on Saturday. So if anyone wants to come along and play this in person, you'll be more than welcome.
Well, thank you for mentioning that. Science Spectacular is a Halloween-themed science fair event happening at SFU Burnaby campus this Saturday, October 29th, from 11 in the morning to 3:30 in the afternoon. So thank you for allowing me to promote that event, and Ben will be at that event. So thank you for mentioning that to everyone in the room. So at this point, I'd like to invite more people to type in questions. I see a few questions on the Q&A box, and I do invite more questions. Keep those questions coming in, and I'll try to read them in the order that we receive these questions. So I'll start with the first one, Ben. When you were at the slide of herd immunity, the question is, what about reinfection? Even if infections are declining, how do you represent people who get infected all over again?
Yeah, so this is a really, really good point. I didn't have quite time to talk about this, but again, it's one of those misconceptions about herd immunity, that it's not this thing that once you achieve it, you just stay there forever. And there could be many reasons as to why you lose herd immunity. So as mentioned, reinfection is one of those. People become reinfected over time because their immunity wanes. And so although you might breach that herd immunity threshold, if immunity wanes over time, as we see with immunity to SARS-CoV-2, as the population loses immunity over time, you can cross back over that below that herd immunity threshold, which allows another epidemic to take off. There are other processes as well, simply births and deaths, as there is known as demographic turnover in a population. As people that have immunity die, through natural causes or through other means, and new individuals are born who are susceptible, that means that, again, your herd immunity threshold could be breached. You could drop below it because of this demographic turnover. We could also see things like evolution of new variants, and that can cause you to drop below your herd immunity threshold. So yes, once you achieve herd immunity, that doesn't mean that you're there and you stay there forever. And that's a really important point that that really needs emphasizing, because I think it tends to be treated as this sort of, you achieve it and then everything's done and everything's fine. Herd immunity is not this static thing that you would just achieve and stay there.
All right. Thank you. Next question. I understand growth, but how do you predict the peak and the disease decreases?
So I understand the growth. How do you predict the? I'm not quite sure I understand the question, but I presume you mean, how do we know exactly when it's going to peak and when it's going to start coming down? We can use in these simple models, we can come up with a mathematical equation that describes what that curve looks like. And based on what that curve looks like, we can say when will the peak occur. These models are what's known as compartmental models. And what they do is they split the population into different compartments, compartmental. And so what we say is, okay, suppose we've got this population and we're going to classify people by their infection status. So these people are classed as susceptible, these people are classed as infected, and these people are classed as recovered and immune. That's an SIR model. And then our mathematical models, all they really do is they talk about the rates of change between those different classes. So people transition from being susceptible to infected at a certain rate, and from infected to recovered at another rate. And there might be more complex dynamics that you could have in there. For example, there could be a latent period, they could have waning immunity and so on. But that's the very simple model. And so we work out when the peak of that epidemic occurs by looking at the balance of what's going into the infected class and what's coming out of the infected class. When those things are perfectly balanced, that means that we're not seeing a change in the number of infections. And so we've seen our epidemic curve rise up as the number of infections is increasing. And then when we see no change in that infection class, the rate of change of that infection class, that means that we're at the peak of our epidemic, and then it will start to come down. So we can use that by within these models and how the rate of infections coming in and going out, how those change and how those become balanced, that will tell us when our peak occurs. Hopefully that answers that question.
Yes, it does. Thank you. Next question. How does mathematical modeling help us predict where the next hotspots will be, or how to predict another outbreak, if at all?
Okay, so it depends if it's a new variant or whether it's so understanding, that's a very good question. So how do we understand new hotspots? So one thing is we can look at how previous infections have played out, maybe. So suppose it's a new variant of COVID-19, and we've seen that it's taken off in another country, say, and we can look at the types of outbreaks that we've had, say, in BC, and then try to say, okay, given the growth rate that we've seen in another country, how does our immunity compare to that country based on prior outbreaks and vaccination levels? And then we can use that to try and predict, okay, how quickly might it grow here? That is quite a difficult thing to do, but in principle, you can do that. And especially as it starts to take off, you can get an idea of how quickly it's maybe growing relative to other things. It's very difficult to maybe predict exactly when a new hotspot is going to emerge and when it's going to emerge. And especially when that comes to new variants, we can't predict what the next variant is going to look like of COVID-19, or we can't predict what the next pandemic is going to look like exactly. We can make some educated guesses about the kinds of things that we would see. So when it comes to COVID-19, I would expect that most of the new variants that we see are being driven by immune escape. That's probably going to be the key thing, and that's the pattern that we've tended to see. Early on, we didn't see much immune escape because there wasn't much immunity in the population. Now there's lots of immunity in the population, and so that's the key thing that tends to drive the evolution of new variants, because they have this big change in their RE number if they're able to escape immunity. But if they don't escape immunity, then a change in transmission doesn't maybe have that big an effect. So yeah, it's very difficult to say exactly when a new hotspot will emerge, or a new virus will emerge, or what the next pandemic might be like. But you can use our models to kind of make educated guesses about the types of dynamics that we'll probably see.
It's not a crystal ball. It's just trying to make the most out of the data we have. Which brings us to the next question. Do you know approximately how many COVID variants there are today?
I mean, it depends. It depends how many, how you define a variant. I guess there's, we tend to think of the Greek letters, as the major variants. There's no, there's no real way, I think, of clearly defining these things. You can take a different definition, based on, okay, a variant has to infect a certain number of people to be classed as a variant. But because the viruses just replicate, they don't reproduce sexually, it's not quite analogous to like species concepts. Even though the concept of species is a whole nother issue that I won't get into today, but that's again something that's not very clearly defined. But the idea of you can't necessarily clearly define what is a variant. They tend to be defined more by the likely impact that they're going to have on the population. So if something looks like it's taking off, or it's particularly virulent, then it maybe gets classed as a new variant of concern. But if we could have hundreds of variants around that don't have what's called different phenotypes, they are perhaps genetically different, but they don't really cause much difference in disease outcomes or who they can infect. We wouldn't probably class them as new variants in the sense of from a public health perspective, because we don't really care about them if they're not causing different outcomes. So there's a little bit of it's a difficult to pin down exactly what the answer to that question is, because the variance is really more determined by the impact on the human population rather than what is a variant from a biological perspective.
Right. Still talking on variants, there's someone in the room who is interested in the initial graphs you had in the earlier part of your presentation that had color saturation at the different parts for the y-axis. What was the significance of those colors?
The initial graphs about the variants had color saturation at different parts of
on the y-axis.
I think I'll just go back. Let me just load up my talk again.
They were different shades of orange, if I remember.
Where was it? I think it was before here.
Earlier part of the slide deck, I think.
There we go. I think it's this one is probably what they're talking about. So what's going on here is the you've got the frequency of the different variants are given by the different colors. So the more, if you imagine each slice, vertical slice is just a moment in time here, a particular date. And the more, if we take, say, November 2021, most of that is this light blue color, and that's the Delta variant. Whereas if we looked at, say, May 2021, there's quite a bit of gray. There's some that's purple, about 40+% of it is this purple, which is the Alpha variant. So I think that question is asking about why do some of these variants go all the way, we see the Omicron variants and the Delta variant going almost to taking up the entire thing, whereas maybe the Alpha variant only takes up about half of this. Again, that's a really interesting and profound question about what's going on in terms of the evolution of the virus and the dynamics within this population. Essentially, the reason that Alpha didn't reach the same levels, didn't reach what's called close to fixation here for compared to Delta or the Omicron variants is because later variants came up, essentially. So Alpha was taking over, and if Delta hadn't emerged, probably would have, swept to fixation, would have taken up, we would have had all of this purple in, summer 2021. But Delta came in and then outcompeted. So Delta was more transmissible than Alpha, and so was able to outcompete it. So that's why we see this transition from Alpha into Delta, because Alpha was fitter than the original type that we had in the population, but then Delta came along and was fitter than Alpha, and so it managed to outcompete Alpha. So it's kind of like competing for resources. We are resources, if you like, for our viruses, and the Delta strain, the Delta variant, was able to outcompete Alpha because it was able to take away more resources from it. So that's why the Alpha variant didn't reach fixation.
Mm-hmm. Thank you. We have a guest in the room who'd like to know your opinion on recombinant variants of a virus and the connected idea of reaching limit to how it can adapt to hosts.
So yeah, viruses are obviously a little bit different from us in terms of how we reproduce. When a virus replicates itself, it's really just usually just making a copy of itself, and when it's undergoing recombination, that's a little bit more like when we reproduce, in a sense, because it's shuffling genes around with other viruses. And that shuffling of genes around can potentially create very different viruses to what you saw before. So typically you'll see point mutations as a virus replicates, and it'll be a little bit different from what it was before. Whereas if you have recombination, it can be very different. And this is particularly important for seeing new strains of things like influenza. So the influenza pandemic of swine flu occurred because a human virus of influenza and an avian virus of influenza both co-occurred in the same pig cell, and there was recombination between those things. And it took a little bit from this one and a little bit from that one and came up with something new that was able to transmit between humans. So recombination can be really important, especially from a pandemic perspective, because it's much more likely to produce something very different to what you've seen before.
Mm-hmm. Thank you. This next one is interesting. How did you get into modeling infectious diseases, or what inspired you to want to study in this field? And maybe for I'm guessing this is a high school student, what are the potential careers in this field?
Yeah, so I was always good at math at school, and that's why I ended up studying mathematics as an undergraduate. But at the time, I didn't know about modeling infectious diseases. I thought, you study math at university to become a math teacher or to go into finance, and those are the career paths. And it wasn't really something that I knew about. So part of the reason I want to do talks like this to the general public is to try to emphasize all the different things that we can use mathematics for, and it's not just going into finance or teaching math. So as an undergraduate, I ended up taking some courses about modeling the biological world. And within that, there was this idea of mathematical epidemiology, and that really captured my inspiration to try to use the tools that I'd learned going through school and through university to understand infectious diseases and how they spread. So the more I started learning about it, thinking initially just about transmission models, and then thinking about evolution, and then thinking about how hosts evolve as well, it kind of built on all of those areas. And as I discovered more, I realized the more that we could use mathematics to understand all of these different things. So yeah, I would thoroughly encourage you to, if you're interested in this and you haven't really thought about mathematics and how it can be used to model things before, mathematics is used throughout evolutionary biology in particular to try to understand how not just infectious diseases evolve, but how different traits might evolve in different populations. Beyond biology, you can use mathematical models in chemistry and physics, economics. There are lots of different ways in which we can use mathematics to model things that we see in the real world. It's not just about necessarily just doing finance or becoming a math teacher.
Thank you. Thank you for that. That's quite inspiring. And because you mentioned it can be applied in different fields, this brings me to the next question. Can you describe any modeling done for social media? Because you mentioned memes in your presentation.
Yes, so I've not looked at any of those papers for quite a long time, but in general, they follow a very similar pattern, as we would model an infection, and that's why I included them here, in the sense that if the sharing rate is high enough, that's just like having a high enough transmission rate for a pathogen, and therefore it can take off exponentially. I think the interesting things, differences with, say, social media, is that you might see things like echo chambers forming that you wouldn't see in quite the same way in infectious disease transmission. And so the key thing with information transmission is information transmission can potentially shape the structure of the contact network in a way that infectious disease transmission can't. And what I mean by that is, we have a network of social contacts, and the information that we share can maybe shape the contacts that we have. That the same kind of thing doesn't happen when we have infectious diseases, where our contact network doesn't really change as much. It's certainly not a long-term contact network really changing much if we have a disease spreading, whereas the information that we share can shape perhaps who we interact with. So I think most of the interesting mathematics and dynamics around that is really about how those networks of contacts, whether it's social media contacts or contacts in the real world as we share information between people, how those networks are shaped by the transmission process itself.
Mm-hmm. Thank you. Next question. Now that a lot of people are traveling and constantly moving, how can this math modeling help us monitor the rate of transmission and the direction of movement of variants?
Yes. So, I mean, it's very difficult to model what's going on globally with transmission. I'd say some interesting things do come out of models when we start thinking about movement between populations at much higher rates. This is one of the things that very simple models can tell us in terms of how do we expect viruses to evolve, for example. So this is a slight deviation from the question, but linked to the idea of international travel, a more connected world means that it's easier for viruses to spread. This means you're much more likely to get an outbreak of a new pathogen because it has access to many more hosts. It's also possible that it might lead to more virulent, more deadly pathogens over time. The reason being that it has access to a much wider range of hosts, and so it doesn't need to, selection doesn't necessarily favor less virulent pathogens in a localized area. So models, it's a little bit complicated to explain here, but the models predict that as you get more and more connected populations, you've got more international travel, you're more likely to get outbreaks that can spread quicker, obviously, but also it can change the dynamics of selection as well, and can potentially lead to more virulent viruses and other pathogens because of changing contact patterns in the population.
Thank you. I think you've touched on this next question, but I'll read it out just the same. Do you think lockdowns were effective?
Yes, I mean, this is something that's quite highly contentious, and I think there's a lot of revisionist history going on, which I'm not going to delve into politics and everything related to lockdowns. But broadly speaking, yes, if you look at countries that went into lockdown earlier and had more stringent lockdowns, we're at a very different stage of the pandemic now. than March 2020. So I think it's important to emphasize that. Lockdowns are your emergency brake. It's not your thing that you want to be doing. It's your thing that you should be doing as a last resort. We don't have any other options. So, shutting down entire economies and schools and health services is not something that I think anyone would want to do. It's really a last resort if things are really going bad. And as I demonstrated in the very simple examples with exponential growth, a short delay in locking down leads to a really bad, exponentially increasing cost. And if you look at the countries that locked down earlier, they tended to have better outcomes. The ones that were stricter with their lockdowns, again, tend to have better outcomes. If you look, for example, at Australia and New Zealand, they tend to keep things under wraps much better, and they had much lower death rates compared to countries like the UK, which delayed its lockdowns and then ended up having longer lockdowns as a result of it and had many more deaths. So overall, it was a blunt measure, and it's very different now compared to back then. So I wouldn't be advocating for lockdowns now unless there was some major change in the virus. We have treatments now that we didn't have before. We have vaccines now that we didn't have before. But broadly speaking, at the time, they were effective. And I think the important thing is not to just think about what's happened in the past, but think about what's going to happen for the next pandemic, because there will be another pandemic. And I think people have tended to think, okay, well, there was Spanish flu in 1918 and COVID-19 was in 2019. And therefore, the next big thing that causes a massive pandemic isn't going to be for another 100 years. These are 1 in 100-year events, and they're not really at all. We've seen multiple viruses emerge into the human population this century alone, whether it's other coronaviruses. We've had SARS-CoV-1 in the early 2000s. We've had swine flu. We've had MERS, we've had avian influenzas coming into the human population. Many of these didn't really take off, but there's always the potential that something's going to take off. And so, I wouldn't be surprised if we have another pandemic in the next 10 or 15 years. It might be longer, it might be less. But the key question is, how do we then respond to that next pandemic? And lockdowns should be something that is a tool in our toolkit. Ideally, we don't have to use it, but at the start of a novel pathogen coming into a population, you don't have treatments and you don't have vaccines. So that really does limit the choices that you can make. And lockdowns can be effective in terms of trying to limit that initial disease spread and prevent your healthcare systems becoming overrun by this exponential growth.
Right. Well, I'll probably end with this last question. And because you did touch a bit on it, but maybe it might be a good way to end tonight's session. How do you think will the next pandemic look like? And what are some examples of behavioral traits that may affect the next pandemic?
Okay, so what do I think it will look like? I mean, if I was a betting man, I would say it's likely to be another respiratory illness. It's going to be probably something like an influenza virus or something like a coronavirus. We've seen coronaviruses, as I mentioned, SARS-CoV-1, SARS-CoV-2. I won't be surprised if there's a SARS-CoV-3 eventually. We've had MERS as well. Various coronaviruses have shown that they've been able to jump into the population. We have these spillover events from animal reservoirs. Likewise, avian influenzas can spill into the human population. They have the thing they have in common is that they are respiratory illnesses. They're very easy to transmit. Unlike something like Ebola, which is highly transmissible, but has very certain characteristics, it's easier to stop the spread of something like that. So I would imagine, what does the next pandemic look like? I would say it's probably going to be a respiratory illness, and I wouldn't be surprised if it's a coronavirus or an influenza virus. In terms of behavioral traits that might affect that, I would say, a key thing from a public health and government sort of top-down perspective is to have better surveillance. We can't, combat or understand what's going to happen if we're not carrying out better surveillance. Something that will also influence this is, human-wildlife interfaces and conflicts. So, the more that we see encroachment from urban into rural areas, the more we see loss of habitat and fragmentation of populations, we might see more interactions between humans and wildlife, which leads to more opportunities for spillover. There are behavioral practices. We talk about things like, wet markets, or any sort of, practices with poultry farms having, opportunities for avian influenza to spread. There are certain, things that we could, how we interact with managed populations for food can affect disease transmission as well. So, yeah, I would say that the behavioral traits that can likely affect this are, what the next pandemic looks like are going to be how we conduct surveillance of possible zoonotic pathogens that could spill into the human population, and the types of practices that might affect that are, often how we interact with either wildlife or food, in terms of, when we have managed populations of poultry or some other species.
Well, thank you. So I guess the key is to understand the information we have on hand and use this to help us plan ahead. So yeah. With that, thank you so much, Ben, for tonight's Café Scientifique. We appreciate your sharing your research with us. And we also want to thank everyone else who joined us in the attendee room. I hope you join us for the next session, November 22nd, save the date, same time, also on a Zoom webinar format. We have someone from our statistics department here at SFU joining us for that evening. I also want to take this opportunity to thank a couple of colleagues we have at our web team. They are here with us. You don't see them, but they are providing our tech support for tonight. Thank you so much for being with us tonight. And thank you, Ben, for always being so wonderful in sharing these presentations with us. Thank you so much, and see you in Science Spectacular this Saturday. And thank you. Well, that's it for tonight. Have a great evening. Thank you all who joined us tonight for tonight's Café Scientifique. I hope to see you on November 22nd. In the meantime, have a great rest of your evening. Take care, everyone.